Hello everyone, welcome to this online session. In this session, we are going to discuss about the drug likeness properties and ADMET properties of molecule, small molecules. So we're going to discuss some of the tools used to predict this drug likeness of a molecules or ADMET properties of a molecule. Okay, so let me share the screen now. We'll discuss in detail about the protein. So first, let us download the drug molecule from the database. Here I'm going to use NCBI database, PubChem database to download a drug molecule. So NCBI databases, we all know that it's major databases for many informations like gene-related, protein-related, metabolic pathway, and the mutation-related many things. And I'm looking for here the drug molecule information. So I choose PubChem compound database. So I choose PubChem compound database, and also I provide the drug of interest here, the theory of interest here. You can see hydroxychloroquine I use. We all know this hydroxychloroquine is highly demanded in India right now, even for this anti-malarial drug. They are using it for corona treatment. Okay. So virus treatment here you can see here when you give one after that you press a search button and you can get the list of um, molecules detail here. There are 13 items we have here in this page. They're all the derivatives, hydroxychloroquine and the molecule here in the first heat. All others are derivatives of the hydroxychloroquine. So I'm going to click on this hydroxychloroquine here. So we will get so many related data here in this page. You can see the 2D structure and 3D structure and all those things. So we need a structure or structural information to study about the drug likeness properties or ADMET properties of this hydroxychloroquine. For that, I'm looking for a three-dimensional structure. So, and also one more important thing I just wanted to tell you here today that canonical smileys. Canonical smileys, it is a simplified format of the compound hydroxychloroquine. We can use either the molecules, the structure for further prediction or analysis, or you can use the canonical smileys of the molecules. Canonical smileys is nothing but simplified molecular input line into specification. You can, I'm going to take the smileys of the molecules. Okay. So I just copy the smileys of the molecule, hydroxychloroquine, that means hydroxychloroquine. So I go to the MolSoft software here for drug likeness properties analysis. You go to the MolSoft LLC, their drug likeness property analysis. One of the best tool I've seen for this analysis, drug likeness properties and everything. When you click on the drug likeness and molecular properties option here, you can see this page where you can upload your drug molecule details here. For example, here we can draw the drug molecule structure here. Yeah, you can using this all these option atomic options and the atomic ring options. You can draw the molecules, or else you can also upload the structures. Yeah. If we click here, it is asking the paste mol or smileys or inch here. So here I'm going to paste the smileys of the compound here. Yeah. When you paste the smileys of the compound, you can see the structure is uploaded here automatically. It will just read the smileys of the molecule. That means simplified format of the molecule. It is constructed here, the structure. Now I just click on the calculate properties. When you click on the calculate properties, you'll be able to see here there are many parameters that are given here. So drug likeness properties and model score also given here. Whether the molecules which you uploaded is drugs or non-drugs. That information you can get it easily from this tool. You can see here there are graphs. You can see the drugs can be here like this, non-drugs can be here. So actually your molecules lies in these actually drug limitations. It is in this point actually you can see in the graph. And drug model scores also very good here. So you can use this as a drug molecules. The other property, let's look at the other properties here, the molecular properties. You can see here the molecular formula, molecular weight, number of hydrogen bond acceptor, donor, and log P value, log S value, polar surface area, molecular value. There are many parameters we have given here. These parameters are very, very important when you check the druggability of the molecules because the drug should obey the Lipschitz rule of 5. The Lipschitz rule of 5 says the molecular weight should be less than 500. Here you can see the molecular weight 335 is given there. And the hydrogen bond donor, hydrogen bond acceptor, and same way the log P value should be less than 5. Here it is given 3.65. That means partition coefficient of the molecules. So based on that, we will be able to know the information of permeability and the druggability and solubility of the drug molecules. So these parameters are very, very important in a drug designing and discovery. So and the drug likeness properties as well, drug likeness model as well. Okay. Okay, now we have discussed about this molecular property analysis and drug-like property predictions. And one more thing we want to discuss here, ADME property prediction, ADMET property predictions. That tool is here. You can see a PKCSM tool. I'm going to use it. So here you can see that ADMET is nothing but absorption, distribution, metabolism, excretion, and toxicity. So these four parameters we're going to study in this session today in detail. That's called ADMET property predictions. The same molecules I'm going to use as an input here in this server, PKCSM server. So one of the best server recently it is launched and we have uh, studied well in this software. And you can see that in here, the software either you can upload your structure or you can also provide your smiley. Smiley's format you can use it. Why they are asking this file to upload here? Sometimes people are working with more number of molecules, like thousands of molecules or lakhs of molecules, sometimes millions of molecules. They work on that to screen the molecules, to shortlist the molecule for particular disease or particular microbial diseases, organisms. Such case, they just upload all the smileys they can upload, put it in one file. They can upload it. It will predict the details of absorption, distribution, metabolism, excretion properties of those molecules and list out. We'll be able to shortlist the molecule based on these parameters. Okay. Here I'm going to use the smileys of the molecule. Smileys we can get it from NC5 Perfume database. We all know this. And you can see I just paste here the hydroxychloroquine mileage here. And there are different parameters given here: absorption and distribution, metabolism, excretion, and toxicity. When I click the absorption, all these absorption properties is predicting the value now. Because in a computational biology or bioinformatics or any molecular modeling studies, we'll get the results in the form of scores, because the softwares and algorithms calculate the uses scoring matrices and algorithms to generate the values. Based on values, we will be able to conclude the results. Here, the water solubility is given here, the predicted value. The permeability is given here, skin permeability, and glycoprotein inhibitor. So all this analysis we can get it. Also, we can get the log P value and the rotatable bond, all this because these permeability and solubility and absorption, everything depends on this value, molecular weight and partition coefficient, number of rotated bond and acceptor. That's the reason why they given this molecular property values as well here. And also they given whether it's a pGlucoprotein inhibitor, yes or no. The pGlucoprotein substrate, yes or no. So all this information we get. If you want to know more about that distribution, you can also paste this smileys here and click on the distribution, and you will get the distribution values like blood-brain barrier permeability, CSN permeability, whether the drug has ability to um, cross the cell membrane and pass through the cell membrane and reaches its target. So all these informations we can get it easily in this tool. It's like actually in silico animal model we say it is. The metabolism details and everything we can just get it. Whether it can be inhibitor or substrate and all these things we can get it here in this tool. So one of the fantastic tools. After that the drug should be eliminated. The drugs once it is orally administered, it has to reach the target and absorb properly. Once it is absorbed, the metabolism must be done properly. After metabolism is done, its proper function is finished, then it starts to be eliminated from our body. If it stays inside the body, then its system it will cause some side effects. It may be a toxicity in system. So that is the reason why. So the renal OCT substrate and the total clearance, all these things we are going to check the absorption, metabolism, excretion, and toxicity studies as well. Sometimes whether it's a carcinogenic genicity or any oral acute toxicity, deadly 50, lethal dose 50, concentration, everything we can check it here in this pharmacokinetic properties. Okay. So this is the one of the best tool I ever seen that the pharmacokinetic to study the pharmacokinetic properties of the drug molecules. Okay. Today we have discussed that how to convert the drug molecules into Smiley's format, and also how to use the MALSOFT for drug likeness properties prediction, and also how to do the ADME property analysis using PKCSM tool. Okay. I hope you enjoy this session. I hope these tools and surveys and information will be helpful to you. Thank you.